Okay. Good afternoon. Welcome to the Connect Science seminar series. A reminder that we're going to post the seminars, the recording on the website that you see on the first slide. And also a reminder to please post your questions in a Q&A and not send it in the chat. And I'd like to introduce our special speaker today, Ron DePignol, who is a past president and distinguished professor at MD Anderson Cancer Center. And he's one of the giants of cancer medicine. He is a member of the National Academy of Sciences and National Academy of Medicine and fellow of the AAS and also the AACR. And he's very well known in the cancer research field. He's also former faculty member at Harvard before he moved to MD Anderson. So many of you may know of his work. So Ron has published some seminal work on dissecting the role of aging and genomic instability in tumorigenesis using creative mouse models and also preclinical studies to better understand tumorigenesis, and then more recently also got into some aging-related diseases that go beyond cancer. And today he will talk about precision genomics and tumor biology. So thank you for joining us, Ron. Looking forward to your presentation.
Thank you very, very much, Nelly. It is wonderful to have a chance to be with you today. I may have left the DFCI, but the DFCI will never leave me. It's always going to be a place where I had tremendous memories, and also much of the science, my career was shaped by my time there, but also many of the people there. I'm really saddened by David Livingston's passing, who I consider to be a scientific mentor and father. And had a profound impact on my career and many others. And then so many friends, Mayor Four, so Jim DiCaprio, Bill Kalin, Linda Chin, Matt Myerson, Barrett Rollins, Jeff Shapiro. We had great postdocs at the time, Bill Sellers and Ned Sharpless and Steve Vartandi. It's just an amazing environment, and it continues to this day. And of course, Andy Aguirre, who was my first graduate student when I arrived at the Dana Farber. So I have many, many fond memories, and I'm just so grateful for the opportunity to share some of the things that we've learned along the way. These are my disclosures, which do not bear relevance to what I'll be talking about today. And this graphic here tries to capture at least on the cancer side. So as Nelly mentioned, we also work on aging, but in cancer, our thinking is really on two levels. One is for some years now, we've been very interested in identifying cancer-specific vulnerabilities through an examination of the composition and the structure of the cancer genome, in part to identify genetic drivers of cancer, also to understand how and whether those genetic aberrations drive malignant progression in a cooperative way, and third, to identify specific vulnerabilities through an examination of the anatomy of the structural aberrations in the genome. I'll be talking about collateral lethality and synthetic essentiality for part of my talk. And much of that interest stemmed from our observation that telomere dysfunction was a major driver of genomic instability. So that got us thinking about genome structure, architecture, and how that could potentially be exploited. In addition, we're first and foremost tumor biologists. We like, tumor physiology, biology. So my lab has been interested in trying to understand not just how these genetic aberrations commandeer the cancer cell intrinsic hallmarks, such as proliferation and apoptosis and so on, but to also understand how they shape the composition of the tumor microenvironment. And moreover, we've come to appreciate that these host cells also provide symbiotic support for the cancer cell, trophic support, not just mere immune sequestration and new blood vessel formation and so on, but really actively instructing the host to grow new blood vessels, to turn off anti-tumor immunity, and even reprogram metabolism in a major way. So these are all concepts and frameworks that we've used to guide the research that we've done. Now, on the cancer genome level, there are 4 concepts which have dominated the way that we think about cancer genetics and genomics. The first concept is oncogene addiction, or we called it tumor maintenance at the time. So Bernie Weinstein coined the term oncogene addiction. And obviously, also Mike Bishop and our lab were the ones to articulate the concept of tumor maintenance, which I think collectively laid the foundation for the identification of driver oncogenes for which targeted therapies could potentially be important, because those driver oncogenes are rate limiting in cancer, despite the backdrop of many other genetic alterations. So while in the 1990s, it was appreciated that oncogenes can drive the development of cancer, we asked the question of whether or not they still serve essential roles in tumor maintenance against the backdrop of all of these other genetic alterations that occurred in the life history of these cancer cells. And so to answer that question directly experimentally, Linda Chin constructed the first inducible oncogene model. And in this experiment, Linda generated a model of malignant melanoma that was engineered with the TET-inducible RAS transgene, was also null for INC-ARF. And she made 2 important observations. First, that RAS was not only important for driving the genesis of melanoma, but it was also remained critical for tumor maintenance. And this model enabled second site suppression genetics, which identified RAS equivalent events, which were predominantly RTKs, which we now know to be very important escape mechanisms for RAS independent growth and in RAS inhibitors, from the work of Andy Aguirre, for example. In addition, there was a second very important observation, and that is when we flipped this RAS switch off, the very first thing that we saw was a collapse in tumor angiogenesis. There was endothelial cell apoptosis. And that taught us that these oncogenes are not simply affecting the cancer cell intrinsic properties, but they're actively instructing the host, in this case, to grow and maintain new blood vessels, to nourish the tumor, and so on. And so this put us on a path to really understand how these oncogenes influence tumor biology and these cancer cell host cell interactions ever since. Now, as I mentioned, there were 4 concepts. The first one, oncogene addiction. The second one is the Hartwell and Friend concept of synthetic lethality, in which 2 pathways converge on a common biological process to regulate a very important biological process, and that the co-extinction of one pathway or the other is tolerable, but the co-extinction leads to a cell lethal situation. And of course, the most celebrated example of this is BRCA deficiency and the activity of PARP inhibitors there, with clinical proof of concept established for this concept. Over 10 years ago, Florian Mueller developed another concept in the lab, and that was this notion that when you delete tumor suppressors, you obviously can't then target those tumor suppressors. But he reasoned that those deletions not only deleted the tumor suppressor gene, but they also deleted the neighboring housekeeping genes. These are genes that were not involved in the pathogenesis of the cancer, but they served cell essential functions. But cancer cells survive because these cancer cells and mammalian cells in general often have multiple paralogs, functionally redundant paralogs. So he reasoned that extinction of that retained paralog would create a cancer cell specific vulnerability. And there are now many examples of this, enolase, malic enzyme, and others that are now in drug development in pharma and will soon enter clinical trials. So this is a concept that I think will expand the pool of cancer targets for us to consider. What I'll spend a little bit of time on is this 4th concept here, and that's the concept of synthetic essentiality. And the idea here is that now that we've sequenced tens of thousands of cancer genomes, we can begin to do statistical analyses to ask the question of whether or not there are genes like gene A that can sometimes be deleted in cancer, but are never deleted in the context of a tumor suppressor gene deletion. And the idea there was, of course, this could simply belie an epistatic relationship, but the other possibility is that maybe gene A becomes an essential effector for that tumor suppressor gene deficiency. And if it's druggable, it could potentially provide a therapeutic target for these tumor suppressor gene deficiencies. And so we embarked on this effort to search for such genes, to look for, first of all, mutually exclusive events. And what we found in the first example was a gene called CHD1, which is sometimes deleted in prostate cancer, also breast cancer and colon cancer, but is rarely, if ever, deleted in PTEN deficient prostate cancers, or even cancers that have mutations in the PI3 kinase pathway. You could see they're roughly mutually exclusive. And so this got us thinking that maybe CHD1 becomes important in the context of PTEN deficiency. So CHD1 caught our attention because it also turned out to be a really interesting gene. It encodes a chromodomain protein, so it activates gene transcription. And the only thing that was really known at the time was that it was important for maintenance of ESL pluripotency. So we got maybe it might be involved in cancer stemness, et cetera. So we went on a study, and I'm just going to summarize work that Dee Zhao did, and it's published. And what she went on to show was that not only was there a mutually exclusive relationship at the genomic level, but that they were directly biochemically linked. So she defined a pathway in which PTEN regulates JSK3 beta, which phosphorylates the degron of CHD1, which then leads through to its ubiquitination and degradation. So when you're intact for PTEN, CHD1 protein is unstable. And when you delete PTEN, it now becomes stable, and it can bind to its chromatin marks, H3K4 trimethylation marks, to turn on hundreds of genes. And through a variety of different strategies, we identified that the most critical genes in cancer for CHD1 in the context of PTEN deficiency were NF-kappa B network genes such as IL-6. And so this was gratifying because Michael Caron had shown that PTEN activates NF-kappa B, and this provided a nice direct biochemical link of PTEN regulation of CHD1 and CHD1's now regulation of the transcription of these NF-kappa B targets, and that allows for prostate cancer progression. So the next thing that we did after defining the relationship, the biochemical relationship between PTEN and CHD1 and its downstream targets, was to understand the biology. And it regulates things like cell proliferation in the cancer cell, et cetera. But one of the interesting things that we saw in vivo was that the major impact wasn't on the cancer cell compartment, but was actually in the tumor microenvironment. Shown here is a classic, this is the Hong Wu or Pier Paolo Pandolfi PTEN deficient prostate cancer, which develops adenocarcinoma, dies within about a year. And what you can see here is that when we now introduce a conditional allele of CHD1, the double knockouts have essentially an upstaged tumor pathology. And if you do Kaplan-Meier analysis, you can see quite readily that while the single knockouts all die, the double knockouts survive. In fact, the few that died were incomplete CRE-mediated deletion of CHD1. So this proves genetically that CHD1 is essential for PTEN deficient tumorigenesis in the prostate. We also showed similar work in breast as well as in colorectal cancer. Now, what is it actually doing? As I mentioned, it regulates many genes, including NF-kappa B, most prominently IL-6. So when we looked at both unbiased transcriptomic analyses and also the tumor pathology, what we were struck with was a real change in the tumor immune composition. And so you can see here at the top panels, that in the PTEN knockout, and prostate cancer is immunologically very cold, and this is reflected in the mouse model as well. And what you see here is a preponderance of granulocytic MDSCs, M2-like macrophages, and a paucity of CD8s. In contrast, the double knockouts, you see a elimination of the granulocytic MDSCs, M2 macrophages, and then you now see an influx of CHD1. So indeed, one of the things that CHD1 is regulating is the tumor immune microenvironment through the regulation of various cytokines, which I'd be happy to discuss. So now, having this one example, we weren't convinced that synthetic essentiality was a really, hardy concept. So we searched for other examples to really substantiate this concept as being a reasonable strategy for the identification of relevant cancer targets. So here we turned our attention to APC deficiency, which is part of the Wnt signaling pathway, which has been really, really hard to drug. So it was one of the reasons why we wanted to see if we can identify downstream effectors of APC deficiency. And we did this in the context of colorectal cancer. Colorectal cancer is a major cancer killer, over 600,000 deaths worldwide, limited therapeutic options, especially for microsatellite stable disease. And then APC in this cancer is the gatekeeper mutation. It is deleted, mutated, and virtually 90% plus of colorectal cancers. It also has two other signature mutations, P53, which is important for telomere dysfunction, chromosomal instability, and KRAS, which is important for progression and actually metastasis maintenance. And also Scott Lowe showed that APC is not only important for genesis, but it remains important for tumor maintenance as well. So it's a very important oncogene that if we can, tumor suppress gene that if we could target the pathway, it would be very helpful. So to generate a mouse model for this disease, Adam Boutine, when he was in the lab, generated a triallelic signature mutation model, APC, P53, and a TET-inducible RAS allele. And with low dose tamoxifen enema, we're able to then generate APC deficient, P53 deficient, and now a DOX-inducible G12D KRAS allele. So the model nicely recapitulates the evolution of the disease all the way through to liver metastasis and lung metastasis. And in this model, it now put us in a position to begin to really define the genetic interactions of APC deficiency with potential downstream effectors. And the key downstream effector for this pathway, which, by the way, is involved in virtually all cancer hallmarks, from metabolism to immune sequestration and more, is that you have now the stabilization and the ability of beta-catenin to turn on its genes through its interaction with the TCF transcription factor. And that then leads to upregulation of many genes, myriad genes involved in the development of cancer. Now, so then what we did is, again, looked at the cancer genomes. We looked for mutually exclusive events, APC wild type versus APC deficient, what genes are present or absent in those settings. We looked at more than just colon cancer and other cancers as well. We also looked at genome scale loss of function screens for Wnt essential genes. And we also looked at downstream transcriptional targets of beta-catenin. And we brought all of these together, and there were only 5 genes that survived the triangulation. And at the top of the list was TDO2. None of the other genes validated. TDO2 is the only one that was validated. So what is TDO2? Turned out to be very interesting, particularly in light of recent clinical trials with IDO inhibitors. So it turns out that TDO2 and IDOs are critical for converting tryptophan to kynurenine. Kynurenine then binds to the AHR receptor and turns on genes that are important for cancer cell proliferation as well as for immune suppression. So it is the critical rate-limiting step in the regulation of tryptophan concentration in the tumor microenvironment. There's a lot of effort to target the IDOs in melanoma, colon cancer, and so on, and those trials unfortunately failed. And the reason why there was such great interest is because kynurenine, through its engagement of the AHR receptor, turns on cancer cell proliferation and also affects virtually all arms of the immune system to suppress tumor immunity. So hence the interest, the idea being that if we can target IDO perhaps and reduce the kynurenine production, that perhaps we could attenuate cancer cell proliferation as well as unleash the power of the immune system. So those trials didn't quite work. And so given the TDO2 result that we found in our genomic analyses, we then turned our attention to see if TDO2 was in fact a downstream essential effector of APC deficiency. So in our mouse model and in human disease as well, we see that we have activated beta-catenin. We see transcriptional upregulation of TDO2. If you look at colloids, for example, you can see that, and these are isogenic lines in which you just knock out APC, you could see that there's a very significant upregulation of TDO2, which is barely negligible in the wild type setting. And IDO is also very lowly expressed. We know that TCF4, it directly binds to the promoter, and it's APC dependent binding as shown here using MC38s that are isogenic wild type APC versus knockout APC. We now have TCF4 binding to the promoter. And also in beta-catenin reporter assays, we can readily see that activated beta-catenin can transcriptionally upregulate TDO2. So TDO2 is a direct transcriptional target of this Wnt signaling pathway via TCF4. And so the question is, how does it actually work? And so, like we do with many things, we do unbiased analyses to try to begin to understand function in vivo. And one of the first things we did was to try to look for genetic evidence that there was a synthetic essential relationship. And so here we used a variety of human cell lines. Very few of them are APC wild type. RKO is one example. And so we CRISPR'd out APC. And you can see here that we knock down TDO2, you see a significant decrease in the viability of these cells. And other cells that are APC knockout are all sensitive to depletion of TDO2. And then we wanted to do murine studies, and we used our model as well as MC38s, which are fortunately APC wild type. This afforded us with the opportunity to CRISPR out APCs, have an isogenic line. We developed several lines, of course. And again, as we saw in the human, the depletion of TDO2 sensitizes these cells to APC deficiency. No effect in the context of APC wild type. Now, these were the genetic studies. We also did pharmacologic studies using a TDO2 inhibitor, and we got the identical results that I'm showing you here. And so the next thing to do was to do a genetic experiment in vivo. And here we did orthotopic transplantation of our MC38 lines with and without APC. And so these two lines here in the middle are MC38's wild type with or without TDO2 knockdown. So TDO2 depletion has no effect on APC wild type MC38s. When you knock out APC, the tumors are more virulent, so the animals succumb to the disease earlier, much more aggressive disease. And despite this aggressiveness, when we now knock down TDO2, we see that there is a marked increase in the survival of these animals. And this is just showing you that when in the context of APC deficiency, but not wild type, depletion of TDO2 dramatically decreases the proliferation and also markedly increases apoptosis in the cancer cells. And in addition, it has an impact on other cells in the tumor as well. And that came to light through unbiased analyses. And so we then began to try to understand, well, what is AHR actually regulating in a TDO2 dependent manner? And so in our APC knockout MC38s, we knocked down TDO2 and then did transcriptomic analyses. And what we found from the pathway analysis was that there were two major hallmarks that arose. One was glycolysis, and the other one was inflammation. And this tracked very nicely with what was known, and that is that AHR regulates cell proliferation. And essentially what it's doing is it's reprogramming metabolism to be anabolic so that it can fuel cell proliferation. And then we had many signatures that conveyed that AHR was also regulating various immune regulatory pathways. And through an analysis of cells that were changing in the tumor microenvironment, as well as cell signatures by CyberSort, we're able to show that when you knock out APC, you see a major increase in tumor-associated macrophages. And in that setting, if you knock down TDO2, you see fewer macrophages. And these macrophages are M2-like. And so this indicated that TDO2, actually through its regulation of CXCL5, a directed transcriptional target of AHR, is able to then recruit tumor-associated macrophages that are of the M2 phenotype. And so then what we did was to do a clinical trial similar to what was done in the human setting, in which we took our MC38s and we used the IDO inhibitor. And in this inhibitor, we're able to show that essentially there was no difference. So the APC knockouts, again, die a little bit sooner, but there was no difference, no statistical difference, genotype dependent or with pharmacologic inhibition of the outcomes of these animals, similar to what we saw in the human clinical trials. In sharp contrast, we see that with the TDO2 inhibitor, APC knockouts die sooner. The wild types are unaffected by the TDO2 inhibitor, but now we see a significant increase in the survival of these animals upon TDO2 inhibition. And on the tumor microenvironment level, what we see is that when you knock out APC, you go from very few macrophages to now lots of macrophages. In addition, you see some CD8s and then a big decrease in CD8s when you knock out APC. And then when you treat with IDO, nothing happens. But in the case of TDO inhibition, you could see now we decrease the macrophages, and now we have an influx in the CD8s that are now found. And now work that we're currently doing is to audit the checkpoint molecules that are on these invading T cells. And we're combining that with a variety of alternative checkpoints and standard checkpoints to see if we can generate more durable responses. Because monotherapy for TDO, while it increased survival, all of the animals still died. So it wasn't sufficient to generate curative or durable responses. So just to summarize, what we found was that the Wnt signaling regulates TCF, which upregulates TDO2, which now generates kynurenine that activates the AHR transcription factor to increase anabolic metabolism and cell proliferation, as well as upregulate various chemokines, most prominently CXCL5, which then binds to CXCR2 receptors on tumor-associated macrophages. Work that I didn't show is that these macrophages not only provide immune sequestration through suppression, but they also secrete Gas6, which Rumi showed binds to AXL and further provides, again, trophic support. It's this symbiotic feedback loop that gets established between these cells that are initially encumbered by the genetic alteration, and you get this circuit that provides additional opportunities for therapeutic intervention at those various levels and so on. So this is just the second example of synthetic essentiality that I think will begin again to expand the opportunities for us for new targets to essentially target in particular tumor suppressor gene deficiencies, which dominate obviously in cancer. Now I'd like to again continue that theme of these interactions between cancer cells and host cells, but do that through our work in pancreas cancer, which was started by Nabil Bartisi and Andy Aguirre, a lifetime ago, and in the lab at Dana Farber. And they made some really important observations that, first of all, RAS drives metabolic reprogramming of cancer cells, but it does so in collaboration with invading immune cells. Even something as fundamental as metabolism requires cooperation with invading cells. And that, as with this new advent of KRAS inhibitors, we're beginning to learn that not only are there cancer cell intrinsic escape mechanisms, but the host, again, can provide escape mechanisms to thwart KRAS inhibitors. So pancreas cancer genetics is dominated by 4 genetic elements, most prominently RAS, which is essentially a rite of passage, and these tumor suppressor genes that Andy and Nabil knocked out and showed that they were really important for progression of these KRAS-initiated neoplasms. Now, what Alec Kimmelman did when he was in the lab, and also Hao Chung Ying, was to generate an inducible allele of RAS in this KRAS, in this PDAC model system. And in this model, they were able to show that KRAS is not only important for genesis, but even with many other genetic alterations, it is essential for tumor maintenance. And so that really inspired a lot of interest in targeting RAS in pancreas cancer because it was such a critical and rate-limiting element, even in fully formed tumors. And that, when it was extinguished, it led to complete regressions of these cancers. And these cancers become metabolically cold, which taught us that one function of RAS is to reprogram anabolic metabolism, predominantly by activating the non-oxidative arm of the pentose phosphate shunt for nucleotide biosynthesis, but also the HBS pathway and glycosylation and so on. So very important in rewiring the anabolic state of these cancer cells. Now, what was striking, and I'm just going to summarize work that PJ did when he was in the lab, was that, we had tried to figure out, well, how is it that KRAS is regulating these glycolysis genes like hexokinase, for example? And we knew that MYC was responsive to KRAS induction in PDAC, but we couldn't figure out how to connect RAS and MYC. So in a completely separate set of studies, we learned that RAS was regulating these cytokine receptors, IL-4 and IL-13. And then we realized that the ligands for those were actually coming from invading CD4 cells that secreted IL-13 and IL-4 to activate these receptors. And PJ went on to show this activates JAK1, STAT6, and STAT6 sits on the upstream enhancer element of MYC to upregulate MYC and turn on glycolysis genes. So even something as fundamental as you think is cancer cell intrinsic as cancer cell metabolism and oncogenes turning on metabolism, this requires the host to close this circuit and drive full-fledged metabolic reprogramming. And if you knock out these CD4s, you see a very significant decrease in glycolysis. Now, of course, when we extinguished KRAS, we were able to then study potential resistant mechanisms to genetic extinction. And in this model, what Avnish Kapoor showed was that about a third of the animals were cured. So all the animals had complete regression initially, but a third of the animals were cured. And a third of the animals had just boring reactivation of the transgene that was now DOX independent. But a third were true escapers. They were ERK negative, and we then studied the cancer genomes to search for what might be the basis for those escape mechanisms. And quite interestingly, half of them were YAP amplified and activated. And this then we showed regulated E2F, and that promoted proliferation of these cells in a KRAS independent manner. In addition, about a quarter of the escapers had RTK activation, and that's very gratifying because Andy Aguirre's recent paper showed that, in fact, RTKs are activated in patients that show escape from KRAS inhibition. But there were a bunch that we couldn't find any genetics. And so we surmised that maybe it might be epigenetic. So that then prompted Ping-Ping Hao. By the way, this data led to, of course, the interest in using KRAS inhibitors, as well as using YAP and TEAD inhibitors because of the YAP amplification. I have a conflict of interest here because one of the companies that I founded is developing TEAD inhibitors. So I think this is going to potentially be a worthwhile complement, either alone or in addition to KRAS inhibition, to generate more durable responses. So what Ping-Ping then did was ask, well, whether or not there's potential epigenetic mechanisms for escape from KRAS. So she took about 300 ORFs and transduced them into these KRAS regulatable PDAC lines and injected them into mice and looked for in vivo escapers. And there was one hit that had 100% penetrance, and that was HDAC5, so histone deacetylase 5. Conferred KRAS independent growth in 100% of cases. Now, what was really interesting about this is that HDAC5 was not able to rescue in vitro growth. And so then we surmised that there may be something from the host that is enabling KRAS independent growth. And so we looked for ligands that might be candidates. We looked at receptors, et cetera. We looked at about a dozen different ligands, and there was one, TGF-beta, that was able to now enable HDAC5 escapers to grow in vitro. And so we then looked at, well, what was the source of the TGF-beta in this KRAS-independent setting? And so we can appreciate from the CyTOF analysis that there's a big difference between KRAS on versus the HDAC5 KRAS off escapers. You could see, again, there's lots of granulocytic MDSCs in the KRAS on. However, in the HDAC5 escapers, those are lost. And it shifts to a macrophage population. And those macrophages are the major source of TGF-beta in the tumor microenvironment in the HDAC5 escaper tumors. And they're so important that if you use cladronate to wipe out the macrophages, those hosts can no longer support tumor formation of the HDAC5 escapers. Now, there were many experiments that Ping-Ping did, and I'm just going to summarize her published work. She, as I mentioned, showed that KRAS that HDAC5 overexpression could allow for KRAS independent growth. She also showed that when you turn down KRAS, HDAC5 is transcriptionally upregulated through mechanisms that we don't know. HDAC5 represses the repressor of cytokines, SOX3. So when HDAC5 goes up, now cytokines go up. And the most prominent cytokine was CCL2 and a bit of CCL7. CCL2, we showed, bind to the CCR2 alpha receptor on macrophages. Those macrophages secrete TGF-beta, which then activates the TGF-beta receptor and signals downstream through the SMAD4 signaling pathway. In fact, if you knock out SMAD4, this mechanism doesn't work. So it suggests or pretends that potentially SMAD4 status in PDAC might dictate the use of this mechanism or not, and you'd anticipate that the SMAD4 deficient individuals might actually have better responses because they have at least one mechanism less to lead to escape, et cetera. And this is also seen, HDAC5 upregulation is also seen in the context of MEK inhibition as well. So this is at least one mechanism that we've seen in vivo in the context of mouse. And if we neutralize CCL2, inhibit CCR2 alpha, neutralize the TGF-beta receptor, or inhibit HDAC5, all of those pharmacologic or genetic interventions is able to thwart this host support mechanism for KRAS independent growth. And I'm just going to show you a complicated experiment, just go through it very quickly, which is what Ping-Ping did was to transiently turn off RAS for about a month and then flip it back on. And a lot of the tumors come back. Only about half of the animals have durable responses. In the other arm, she kept RAS on. And she tried all of these inhibitors, CCL2, HDAC5, and CCL2 neutralizing antibodies, et cetera. And all of these bars here are the KRAS on with these agents that I just mentioned. And if anything, they did worse in the context of KRAS on. This black line here is just KRAS off in vehicle. And all of the others show that there's now a survival benefit. More animals have durable responses if we now layer in any of these agents that are targeting this circuit that enables KRAS independent growth. And so this at least provides another opportunity for us to utilize agents that have, some of which have been into clinical testing already, to see if they can further enhance the effectiveness of KRAS inhibitors in the context of pancreas cancer. So now I want to just end my talk in talking about one other aspect, which is tumor immunity. And of course, this thinking was dominated by Jim Allison, Gordon Freeman, Arlene Sharp, Hanjo, a truly groundbreaking, seminal, life-saving work in harnessing the power of the immune system. And we are forever grateful for their important advances. As you may know, PDAC is perhaps the most or one of the most immunologically cold tumors. And so Pat Gulhati, who was then a postdoc in the lab, now at Rutgers, asked the question, why is it so immunologically cold? And are there cracks in the armor that we can identify where we can systematically study these immune mechanisms and target them one by one or in combination? And as I mentioned, PDAC is a really non-responsive tumor. It's essentially a brick when it comes to IO, and very few responses with, the first wave IO therapies developed by Gordon, for example, as well as by Jim. And even in combination, very few patients responded. And so what Pat hypothesized is that maybe it's not CTLA-4 or PD-1, but maybe some of these other alternative checkpoints like LAG-3 or VISTA, TIM-3, or these agonist receptors. And then the other possibility from our work and others was the importance of myeloid cells. And they are, in fact, I think, dominant. While we've done a lot of work on checkpoint over the last decade, I think this next decade is going to be dominated by a lot of targeting of myeloid checkpoints. And so what he did was to do auditing of the molecules that are found in both human and mouse. And I'm just showing you the bottom line here, which is that in the single cell sequencing analysis that we did with Nick Navin, we found that in the exhausted CD8s, there were PD-1, LAG-3, 4-1BB, and TIM-3. If you look at the developmental trajectory from naive to activated to exhausted, you could see LAG-3, PD-1, 4-1BB, CTLA-4, OX40, TIM-3 are the players. And so he then embarked on an effort to do clinical trials to target these. Now, in addition, he also showed through looking at naive or treated pancreas cancer patients that in particular, LAG-3 and 4-1BB were highly represented on the T cells in the majority of pancreas cancer patients. And this is just single cell sequencing data from the references that are shown at the bottom, showing that LAG-3 and 4-1BB are indeed expressed in the T cells that are resident within the tumor microenvironment in human pancreas cancer. So then he did a study in which he treated the animals for 4 weeks with monotherapy using directed against those T cell checkpoints. He did a single dose of gemcitabine, which is the standard of care also to induce immunologic cell death, and then did interim analysis and survival analysis. On the interim analysis, what he showed was that PD-1 CTLA-4 didn't work, as similar to what we saw in the clinic. However, when we now look at LAG-3 or 4-1BB, we see a reduction in tumor growth. And there's also an increase in the survival of those animals that were treated with antagonistic LAG-3 or agonistic 4-1BB. And then we did single cell sequencing analysis. You could see how large these tumors were at the time of treatment. We did this again with Nick Navin and Aislinn in his lab to really begin to understand, well, what are the adaptive mechanisms in the myeloid compartment or other T cell checkpoints, et cetera, that are happening in the context of this monotherapy and so on. And what we can readily see is that there was clearly a T cell activation response with the successful LAG3 and 4-1BB, but not in the context of control or PD-1s or CTLA-4, which did not have much in the way of a T cell receptor response. But you can see here that there's diversification of the T cell receptor in the context of the effective monotherapies for 4-1BB and LAG3. And so the next thing he did, since 4-1BB and LAG3 work through different mechanisms, was to combine them to see if you can get additive or even curative effects. And what he found was that they were indeed additive, as we anticipated, but none of the animals developed tail, suggesting more durable responses. So we then turned our attention to the myeloid compartment. And the reason for that is that there is a preponderance of granulocytic and some monocytic MDSCs in the mouse, and a signature we developed years ago to look at an equivalent population in human. We were able to discern an equivalent granulocytic MDSC population in human pancreas cancer as well. And so, in fact, if we take that signature, more than any other immune signature, MDSC signature, high and low, is the most important correlative outcome in human pancreas cancer. And these cells express very high levels of CXCR1 and 2. And so that then prompted us to do a study using CX1R2 inhibitors. And what we found was that with monotherapy, the Syntrex inhibitor SX682 is able to decrease tumor volume, is able to increase survival, but again, no durable responses. But now in triple therapy, so LAG3 agonist, antagonist, 4-1B agonist, and the small molecule inhibitor for CXCR1-2, we see that about 90% of the animals are cured of their disease. They're eradicated. They live. At necropsy, there was no evidence of disease. So this tells us that pancreas cancer is, in fact, amenable to immune therapy, but it requires multiple targeted therapies in order to really generate these kinds of durable responses. Now, we see that, as would be expected with the combination, a very nice increase in effector T cells. We also are able to show that we can induce immunologic cell memory. So previously treated animals, when re-injected with the cancer cells, eradicate those cancer cells and survive, whereas the naive treated controls were able to succumb to injection of these cancer cells. And then just a balloon on the horizon is that, when we are seeing adaptive mechanisms, not all of the animals are cured. And again, these, immune systems are highly malleable. And what we're seeing is that these granulocytic MDSCs are eliminated by the Syntrex inhibitor, but we see the emergence now of monocytic MDSCs. And so we're now targeting those to see if that could also lead to more durable responses. But I just wanted to bring this to your attention, that this is another potential therapeutic target for us. I'll mention that the triple therapy was tolerated. There was transient elevation of LFTs, which was seen with the LAG3 inhibitor, but it returned to normal after a dose or two. But the animals did quite well, actually, for throughout the course of treatment for these animals. And then finally, the highest bar, orthochronous tumors. Nothing works in this model. We've been working on it for 20 years. We see that with sustained treatment, we see that the triple therapy is able to lead to a very significant increase in survival. And you should understand that in the orthochronous tumor, the entire pancreas essentially becomes a cancer. There are multifocal cancers that arise. So these are large, highly aggressive tumors. And here we can see that the triple therapy does provide survival benefit. And about 20% of the animals at 6 months at necropsy had no evidence of disease. And so whether or not they're truly cured, we don't know. But it looks good that they at least eradicated their disease to that point and would have survived at least for some time thereafter. But we sacrificed them to finish up this story for publication. So just to summarize, we see CD8s in T cells in the tumor microenvironment. This, along with the myeloid cells, suggested potential for targeting alternative checkpoints in the myeloid compartment. We did a comprehensive analysis using a variety of omic studies as well as immune profiling, leading to the identification of LAG3, 401BB, and CXCR12 as critical targets. It was well tolerated, and all of these molecules are expressed in human PDAC specimens. All of these molecules have been in clinical trials, so it provides opportunities for us to really potentially take these drugs and begin to combine them. And hopefully some of these trials, combination trials, will be underway by the end of the year. So I just want to end by saying that, by looking at the cancer genome, we've been able to identify not just driver oncogenes, but ways of targeting tumor suppressor genes through collateral lethal as well as synthetic essential approaches. And also we've come to appreciate over the last 20 years that these oncogenic events not only commandeer cancer cell intrinsic properties, but they play a direct and proactive role in shaping the immune microenvironment and the kinds of circuits. Different oncogenes, they bring in different cells or the same cells, but through different cytokines, different receptors, et cetera, highly complex. And these cells in turn provide support for the developing cancer cell as well. And so again, providing opportunities, not just for us to target the cancer cell compartment, not just unleash the power of the immune system, but to target these symbiotic interactions, heterotypic interactions, and growth factors that are enabling this interaction with the cancer. These are the folks that did the work over the years. So, a lot of the folks who helped launch the PDAC program in particular. We've had amazing collaborators over the years. The recent work that we've done on pancreas cancer immunotherapy was enabled by lots of folks in the lab, as well as those here at MD Anderson, like Michael Kim and Huamin Wang, who provided critical human specimens. We were helped on the omics side by Nick and Aislinn on a single cell, reagents from Syntrex, and biostatistical support from our colleagues at Rutgers. Thank you very much for giving me the opportunity to present today.
Okay, thank you, Ron. That was very impressive. Really, really exciting story. It looks like everything leads to immunology. So we have a couple of questions here. I can start reading them. One of the questions is there any link between TDO2, APC, and HIF1 alpha or HIF2 because there is an increase in hypoxia signature?
Yeah, we did not specifically look at that in the context of TDO2, but one of the other factors that are binding elements that are responsive to KRAS and PDAC is HIF. But we did not specifically look at HIF and figure out the circuitry there, et cetera. We focused our attention solely on MYC.
Mm-hmm. Okay, another question is APC loss, KRAS mutation, and P53 loss sufficient to drive liver metastasis?
Basically, so APC and P53 on a genome stable background, so not telomere dysfunction, right? That can drive local tumor growth. APC plays an important role in the maintenance of primary tumors, Scott Lowe's work. Yeah. What we showed was that RAS is critical, or RAS equivalent events are critical for progression, invasion, and metastasis maintenance. So if you flip off RAS, you eliminate the invasive and the metastatic disease. And the mechanism there is through RAS repression of IRF2. So IRF2 regulates cytokines that regulate both EMT as well as immune mechanisms. And in fact, it's so important in colon cancer that deletion of IRF2 and mutation of KRAS are mutually exclusive. And so there's an epistatic relationship, even at the genomic level and in human pancreas cancer. So those are the three mechanisms. And what we're now doing is we've now created a mouse model for genome instability in the context of this model, which will enable us to more faithfully, model the really key aspects of the disease, which is APC, P53, a double-strand break-inducing process, namely telomere dysfunction, as well as RAS. And so that together will give us, opportunities to study gene function, discover new genes, but also really understand how that's responding to therapies and so on.
Mm-hmm. Right. Another question. What is the role of mitochondria in T-cell exhaustion and senescence?
Wow. Okay. Well, that's another lecture.
Yeah.
Mitochondria, obviously figure very, very, very prominently in this process. So not only in metabolic processes and so on, but particularly in the context of genomics and genomic instability, through a pathway we discovered, P53 regulating Bruce Spiegelman's favorite, molecules, PGC1 alpha and beta, that plays a really critical role in mitochondrial biogenesis and virtually everything relating to mitochondrial function, and regulating oxidative defense in a major way, because they regulate virtually all the oxidative defense molecules and so on. So there is a absolute intimate link between these signaling pathways and P53 in the context of mitochondrial biology and oxidative defense. And those also have very significant impact on the immune responses, et cetera, through mechanisms that involve things like seagasting, et cetera. So it's a very important and integral part of this whole story. And that's really almost a whole other lecture that we give in the context of telomere dysfunction.
Mm-hmm. Okay. And Ron, so this idea about the, the FAP, the APC mutant cells being dependent on TDO2. So raises the question in FAP patients, that are heterozygous, is there any phenotype that could be related? Like, is there something different in the immune environment or anything?
We're just looking at that now. And there are some other very interesting things beyond what I presented that was recently in the literature showing that APC, and I think that this is going to be important for APC patients, that one of the other critical functions that early APC lesion does is to upregulate Wnt antagonists to clear the microenvironment of other normal stem cells and inhibit and create a niche, an advantage and a niche to allow. So I think there's a lot of things that we can now study in FAP at the mechanistic level from the work of others, but also really trying to understand whether or not there are unique tumor immune microenvironments that get encumbered because of the genetics up front. And not in the context of APC, it's almost a rite of passage for colon cancer, but some, will target Ramesh Sivdasani's favorite molecule, CDX2, as a way of dysregulating the Wnt signaling pathway and so on. And so it'd be very interesting to look at the immune architecture, in that setting. And APC obviously is a really important model for APC, FAP.
Okay. Yeah. Okay. Few more questions. Are you concerned that the relative responses in mouse can fail to translate to humans because the cell numbers and, in humans, you have more clonal diversity, so higher risk of escape?
Yeah, absolutely, unequivocally true.
Yeah.
I will say that in the last 20 years, over 100 agents have been used in this model, and just like in humans, they didn't work. And so, at least at that level, it's just nicely recapitulated, unfortunately, what we saw on the human side, non-responsive CTLA-4, non-responsive PD-1, and so on. I think what this teaches us is that it's something now to test the clinical hypothesis.
Mhm.
And the fact that these molecules are found in human, the fact that you see correlations with outcome, the myeloid signature that I showed, I think is something we should pay attention to. That's number one. Number two is in the context of prostate cancer, we actually showed that in that case, dual checkpoint blockade, CTLA-4 and PD-1, together with the CXCR2 inhibitor in P10, SMAD4, and P53 deficient prostate cancer, castrate-resistant, treated with enzalutamide, metastatic bony disease. Those animals were cured with the combination of the dual checkpoint blockade and molecules that targeted myeloid suppressor cells. Now, just recently, those clinical trials have now been initiated, and one of the agents we used was Cabo, which preferentially targets MDSCs and spares T cells, and they're seeing nice responses in prostate cancer with Cabo and PD-1. And so, it's just, these are just provide hypotheses. And generally, if it doesn't work in the mouse, it won't work in human. But humans do have genomic instability, which most mouse models do not recapitulate, which we know to be critically important for escape, for immune modulation, and so on.
Okay. Well, thank you. We don't have time for more questions, but we can save those and forward it to you. So thank you again for the audience for joining us today. Thank you, Ron, for a very impressive talk. And again, the recording will be available in the website probably by tomorrow if somebody would like to look at it again. All right. Thank you. Bye-bye.